Hi guys, let's study with me and let's study the fundamentals of microbiology. This course will try to improve your fundamentals and watch this video for as an introduction to microbiology. Let's start. So the fundamental question before anything is to ask the right question, right? So the wrong question is that when someone hands you an unknown microbe and the instinct is to ask, is it staph, TB, or what is it? Is it Candida? That's guessing. You should try to classify. Now the thing is, how do you want to classify the whole subject, the whole microbiology, right? It's a big subject. Well, we can first ask a simple question that is it alive or is it dead? If it's not alive, then we can think it's a virus. And if it's alive, then we can think, okay, it may be a bacterium, it may be a fungus, or it can be a parasite. Why it matters? Because a single fork dividing all the microbiology. So alive and dead. Viruses are dead and rest of them are alive, right? Let's ask the question, if something can copy itself inside another cell, is it alive or dead? It's a good question. Borderline. This is exactly a virus is, okay? It carries this information but cannot run its own metabolism. It needs this metabolism for running its DNA, right? So by strict definition, it sits on the not alive part, okay? In the kingdom of microbiology, we have 5 great branches, okay, that root from one thing. That we have microbiology, we have bacteria, we have viruses, fungi, parasites, protozoa, helminths under the parasite, and then we have prions. Let's start simply understanding them one by one, okay? So these branches are alive bacteria, okay? All of them are from alive. All of them, they include alive bacteria. Now, 2 are not, okay? Viruses and prions. Viruses and prions, these are actually dead, kind of dead. So parasites split into 2 cells, right? Protozoa and helminths. Now we want to ask ourselves, where do the worms sit on the tree? Worm is alive, so what do you think? Well, it is under parasite. So helminths, these are worms. They are eukaryote, means they are quite advanced, right? Eukaryote means they are quite advanced. Prokaryote means they are quite old. These are multicellular. It means they are quite complex. They are quite gifted by nature, and they are very much alive, right? Now we ask a simple question: what is life? Well, 5 jobs separates the living from the inert, right? Inert is just dead. So biologically, to be alive is to grow, metabolize, and produce, make its own proteins, and produce the ATP, the energy. So these are the 5 things that you want to do if you're alive, right? So bacteria can do it, fungi can do it, parasites can do it as well, but virus cannot do most of it, okay? And prions cannot do anything. Prions is absolutely dead. So why viruses are mostly not alive? Because they store information and they can direct their own copying machinery, but they borrow their host machinery to do it, right? They have their own DNA, but they need our metabolism to work, to be alive. So no independent metabolism or energy production as far as I know. So they fail the test of being alive. Let's talk about prions. So what are prions? Prions are super duper strange. Prion is an infection protein, right? It's just a protein. It's not a DNA, it's not an RNA, and they don't have any metabolism. They have nothing going on except a badly folded protein, right? It's a bad shaped protein, and this is how the infection itself happens, right? The folding cascade. Picture one badly folded shirt touching another one. Now both of them are folded wrongly, and then next one gets misfolded, and then next one. The bad shape templates creates more bad shape, just like the animation right in front of your eyes right now. So we have 2 diseases, Creutzfeldt and Jakob. These 2 diseases are super duper tough. These are untreatable, and you are gonna eventually die if you have this. So they are resistant to ordinary sterilization as well. So that is super duper scary. I'm actually quite afraid of prions myself. So if a prion has no genome, how can it be infectious? That's a good question, no? Well, it propagates a shape, okay, not a sequence. The misfolded protein converts your normal copies into misfolded form, and a chain of reaction confirmation happens, and that's not a replication of code, that's a replication of shape. So it's literally bad company. Viruses are like this genetic USB that goes inside your host cell and then use your metabolism to actually mess you up. So a virus is not a cell, it is a information plus protective coat, right? And then this coat can be present in different shapes and sizes, okay? Nothing else. Think of this viruses as a USB stick. On its own, it does nothing. Plug it into a computer, suddenly it copies itself everywhere. A virus is same, inert until it reaches the host. Now we want to ask ourselves a question: what does the virus borrows? What does the virus borrows from the host? Energy, metabolism, entire factory, ribosome, energy, building blocks. The virus supplies only the instruction, right? The host does the all work of copying. That is so super duper weird, I think, but it really does works like this. Virus structure. Now the virus structure has genome, a capsid, and an optional stolen code. Now it is a stolen code, remember that. Every virus carries a genome. A genome is DNA or an RNA, right? DNA has two sugar phosphate backbones, and RNA has a single strand, right? Usually, never both. That is very important. This information is the code, okay? It will not have both DNA and RNA. It will either have a DNA or it will have an RNA. And the capsid. What is capsid? Around it sits the capsid. Now what is capsid? A protein shell that protects the genome and helps the virus to attach to a cell, right? So this is a genome, right? This spring, do you see it? The spring right there, right? That golden thing protecting the spring, the DNA, the genome, the DNA is the capsid itself, right? Now the viruses can be enveloped or it can have spikes as well, right? Let's see the envelope. Some viruses add another envelope. This is a lipid, means it's a fat. It's a fat membrane stolen from the host, studded with spike protein. So viruses is genome, capsid, plus maybe envelope, maybe not envelope, okay? So virus can be DNA or RNA, okay? And the shape of these genome can be actually different. They can be circular, they can be spring, many type of different genomes. Now, why does the DNA or RNA, but never both, matter clinically? Why? Because it is the first sorting question for any virus. The nucleic acid type predicts the mutation rate, replication site, and drug strategy. A lot of viruses, they mutate really fast, so it's really tough for us to make any sort of vaccine for them. But some viruses, they do not mutate as much as other viruses, so it is much easier for us to have vaccines for them. DNA virus versus RNA virus. Cool question. So DNA virus has two strands, as you can see, two strands, and RNA virus only has one strand, right? That's the strand. Now, two temperatures. DNA viruses are stable, mutate slowly, and usually replicate in the nucleus. It's a very important word. RNA viruses are usually unstable, mutate fast, and usually work in the cytoplasm because RNA usually feeds into where? Do you know where? So RNA virus actually works in cytosol because we have this machinery, this complex machinery present in the cytosol. So of course RNA virus is going to work in cytosol. RNA viruses mutate fast because RNA polymerase has a poor proofreading. Of course, it's not DNA polymerase. You know the error rate of DNA polymerase. The error rate of DNA polymerase is around 1 to 1 billion. So it copies around 1 billion, these AG, AT, these base pairs, and after copying like 1 billion base pairs, it makes like one mistake. So that's how efficient our DNA polymerase is, but RNA polymerase is not really super efficient, so it makes a lot of mistakes. So this is why influenza and HIV they evolve super duper quickly. Now we ask ourselves, why do RNA viruses make better vaccine evasion artists, right? Because they mutate so fast. They are RNA. They don't have a very stable genome. RNA is actually great for transcription. So not transcription, translation. So RNA is super duper volatile. So if a virus is RNA, so of course it's gonna mutate super duper fast. Next question we want to ask ourselves about viruses is that virus enveloped or is that naked? Because enveloped virus surprisingly are soft and are destroyed by alcohol, soap, and drying and heat. But naked viruses are super duper tough. Now you would think that logically, something is enveloped. It must be tough to be destroyed. But actually, contrary to your belief, you don't need much to destroy these things, destroy these enveloped viruses. But naked viruses are super duper tough. They do not die. Trust me. So not naked viruses are tough. They survive in acid stomach, right? Naked, they can survive inside our acid. So fecal oral, enveloped, they die easily. So respiratory, sexual, blood. So just clean your hands, use soap, use some alcohol, right? They'll disappear. So why does hand sanitizer work on flu but does not on norovirus? Because flu is enveloped, right? Alcohol melts its lipid coat. Norovirus is super duper naked. Norovirus is super duper famous on the these. It's very infectious basically. Norovirus usually messes up with the children, and it gives some diarrhea and this kind of GIT problems. So it's hard for norovirus is naked, so it's hard protein capsid shrugs off the alcohol, right? It has a protein capsid, and capsid is basically a covering. Okay, so you need soap and water or bleach to really deal with it. Now let's talk about the shape. I already told you shape is very important. Now we have three types of shape. First shape is icosahedral. This is a soccer ball, right? This is icosahedral, a soccer ball. And then we have helical. Now helical is like how DNA looks like, right? Helical, spiral protein. And complex is an alien spaceship. This is super duper weird. It is scary and it is weird. Alien spaceship like a bacteriophage and with a head and a tail. This is scary. But it's complex. It's a phage, isospiral, and icosahedral. Let's talk why shape matters. Because shape will decide the type of attachment the virus was gonna show, and shape will decide the type of pathogenesis the virus will show. So how a virus is built decides what it grabs and how it gets in, of course. So which shape lands its genome like a syringe? Do you think so? What do you think? Of course, the complex bacteriophage. Its tail fibers anchor to the host and sheath injects the genome directly. So this is insane. These teeth actually dig into your membrane and then we have a syringe, right? So that's how it works. Okay, let's come to the bacteria now. So we are done with viruses. Let's talk about bacteria. Bacteria are super duper alive. They are alive and well. They're tiny, they're complete, they are self-sufficient. They are living. So bacteria eat, grow, divide, make protein, produce ADP, right? Unlike viruses, they do not need any sort of host. They are a living organism in its own, right? They produce by binary fission, right? Now the cell elongates. The binary fission is happening like this, okay? In the animation, if you look at it. So it copies its DNA, pinches out in the middle, and then splits into two identical cells. This is how literally it happens. Everything starts here. So almost everything a bacterium does traces back to one structure, its cell wall. Now hold that thought. So we can divide the bacteria very well depending upon the cell wall, okay? Why is binary fission so dangerous clinically? Because it is exponential. Now, one cell, right? It is super duper tough because it happens a lot. They will have a lot of reproduction. So one cell becomes 2, then 2 becomes 4. Under good condition, a population can double every 20 minutes. That is very overwhelming, right? So I told you, hold that idea of cell wall because we can actually divide the bacteria into many types depending on the cell wall. Now, bacteria cell wall, 5 layers memorized from inside out. So the most inside is the DNA. We have nucleoid, right? Then we have cytoplasm, right? Then we have cell membrane, cell wall, and capsule. Now this order is important because everything downstream, staining, antibiotic, virulence, depends upon which layer is present, right? So we want to memorize the outward sequence forever, the capsule, the cell wall, okay? This is how it looks like. So which layer is not present in every bacterium? That's a good way to categorize. Which way? The capsule. The capsule is optional. And a virulence bonus, right? Capsule is an option. It's optional and it's a virulence bonus, right? The membrane, the walls, cytoplasm, and DNA are essential to every bacteria. So most of the bacteria can be divided on capsule, if they have the capsule or not. Usually, capsulated bacteria are super duper tough to catch. So you need these opsonization, right, by your C3b or by your IgG, and you need this opsonization. Opsonization is just basically trying to catch the capsules, and your spleen is usually responsible for catching these capsulated bacteria. If you don't have spleen, maybe due to spherocytosis or maybe some sickle cells, you are cooked. you have problems. Okay, the capsule. Invisible slime armor. The capsule is slippery outer coat. Now its job is to protect the bacterium from phagocytosis and opsonization and drying and immune attack. Of course, it is super duper slippery. Now, more slim, more dangerous. A bigger capsule means more virulence. Of course, classic encapsulated bacteria are Staphylococcus pneumoniae, Klebsiella pneumoniae, and H. influenza type B. These are all encapsulated. Now we're going to ask ourselves, how do vaccines exploit this capsule? How are we going to manipulate this? Well, simply many vaccines like pneumococcal and HIV meningococcal target capsular sugars, the sugar that is how the capsule is made up of, right? So what's going to happen is that we're going to target that capsular sugar, and we're going to take some little part of that capsule, and we're going to teach our immune system to grab that very coat every time it sees. So normally that bacteria will not slip away after teaching an immune system, right? So the cell wall. Cell wall is the basic. The cell wall is the way we wanna stain your bacteria, right? So cell wall, the structure that decides everything. The cell wall determines the gram stain, the shape, and how the antibiotics work. When the bacterium virulence, and everything radiates from here. So cell wall is super duper important because exactly bacteria, virulence, protection, gram stain, right? Everything depends upon cell wall. Now, one structure and many answers. Of course, when a wall question feels like 5 separate facts, remember that they are just one fact wearing 5 different hats, right? So we want to understand the cell wall on a basic level. Now, first, let's talk about antibiotics. So let's talk about penicillin. Now, first question is, why do penicillin do not harm human cells? Because human cells have no peptidoglycan wall. Now we are talking about a peptidoglycan wall. Peptido means protein, glycan means sugar. So sugar protein wall. Now penicillin attacks this peptidoglycan wall building enzyme, the enzyme that builds this peptidoglycan wall. Now if we don't have that peptidoglycan wall, the penicillin is not going to do much to us, right? So it's like a perfect selective target. Now, peptidoglycan, right? The big ball that stops the cell from bursting, right? Peptidoglycan is a mesh of sugar. Which sugar? NAG and NAM, 2 sugars, right? They're locked with peptidoglycan cross-linking. So this is the cross-linking. This is NAG, this is NAM. And peptide cross-linking is happening right here in the animation, right? So what really happens here is that bacteria sits in a trigger pressure that would burst them, right? The peptidoglycan mesh is strong enough to hold this cell wall intact. Without it, the bacteria itself will explode. Because if you remove the structural integrity of the bacteria, how the hell do you think bacteria is gonna survive, right? So what happens when the antibiotic blocks this cross-linking? What's gonna happen? The wall is gonna be super weak. That's what's gonna happen. The mesh can't hold the pressure. Water rushes in. The cell lysis happens. Cell lysis means cell death, and this is how precisely beta-lactam kills. Now, beta-lactam. What is beta-lactam? First of all, I just wrote that. Beta-lactam is, for example, we gave amoxicillin clovenate. Now that clovenate part is the protector of beta-lactam, or am I wrong? Maybe I'm wrong. Okay, never mind. Okay, so gram stain. Let's talk about gram stain. The single most important classification, of course. Now, basically we are trying to ask ourselves that, hey, does the bacteria have thick wall or thin wall? Basically, that's the question. So gram stain, how thick is the peptidoglycan wall? So thick is gram positive, that is purple. Thin is gram negative, that is pink or red, or whatever, depending on what you want to know or what you want to think. So crystal violet stains both, of course. So both of them are going to be violet. So alcohol decolorizes them. Yeah, once you put both of them in alcohol, both of them decolorize. And the thin wall loses its dye and the thick wall keeps it, because thick wall has a lot of peptidoglycan, and peptidoglycan loves this stain. So that's why. Other thing is saffron then turns the thin one into pink. We dip both of them in saffron, and then the thin one actually loves saffron. So this is basically thin. Gram negative is thin wall, gram positive is thick wall. Simple stuff. So half the subject. Now half of the clinical microbiology branches this into one result. It's the first thing you want to ask in cultured bacterium. Yeah, it's true. Why does the thick wall trap crystal violet? Because it has peptidoglycan. Alcohol dehydrates and shrinks the dense peptidoglycan, locking the dye inside. The thin wall gram negative have an outer membrane that alcohol dissolves, washing the dye out. I hope you understand this explanation. Okay, gram positive, the thick wall, right? So thick peptidoglycan threaded with teichoic acid and no outer membrane. This word is very important, no outer membrane. I will tell you why. So we have these acids right there, these little sticks pointing inside the thick membrane. The teichoic acid matters for adhesion, inflammation, and antigenicity. Now, Staph, Strep, Bacillus, Clostridium. These are the example of gram-positive. Why are gram-positive generally more vulnerable to penicillin G? That's a nice question, right? Because their thick peptidoglycan is fully exposed. They do not have any outer membrane, right? So no outer membrane shields it, so the drug reaches its target really fast, really directly. Gram negative. So we have gram negative bacteria. So thin peptidoglycan wall, and we're gonna have an outer membrane that's gonna protect this bacteria from penicillin G. That's very important. So thin wall, extra membrane, and outer membrane over thin peptidoglycan. They do not have those acids which were present, which teichoic acid. Gram negative have something else. They have LPS. What is LPS? LPS is an endotoxin. Now, it's not an exotoxin, it's an endotoxin. Why it matters? LPS is an endotoxin. Lipopolysaccharides is basically fat and sugar, basically. And wait, I'll continue. Yeah, I'm back. So we were talking about LPS. So LPS is a it's basically a protein and sugar, right? And what happens here is that we have this molecule, and it is the reason we have this inflammation after a bacterial infection. That's the reason. And we have this septic shock and hypotension. It's super duper toxic and super duper dangerous. Okay. Why are gram negative often harder to treat? Because they have this outer covering, right? So it's a problem. Okay, let me see what's the answer. The outer membrane is the barrier that blocks many drugs, and its LPS is dangerous to sepsis. Two problems, one envelope. Of course. So let's talk about endotoxin itself, the LPS, okay? So endotoxin, it has three parts, and it's a villain, right? So we have lipid A, we have core polysaccharide itself, and then we have an O antigen, right? So we have a lipid A, that's the toxic part. The lipid A is the toxic part itself. Then we have a core polysaccharide. Now this core polysaccharide is just a sugar, and poly means just a lot, and saccharide is just sugar, so a lot of. And then we have a O antigen. Now this is O antigen is super duper important because we use that O antigen to understand what type of strain it is, right? So lipid A triggers the cascade, fever, septic shock, hypotension. Remember, this is the main thing. What antibiotics sometimes people can take to make sepsis worse? This is a cool, super duper cool answer. When we kill the gram-negative bacteria, it releases a lot of LPS, right? That is a transient surge of endotoxin. For example, E. coli. Okay, so exotoxin, one of my favorite thing, because a lot of people, they end up giving antibiotics to the endotoxin, but we don't want to do that. We do not want to do that. Yes. So exotoxin, secreted precision weapon. So secreted poison, right? An exotoxin is just a toxin that is secreted exogenously, right? So an exotoxin is secreted in protein, powerful, highly specific. So botulinum, cholera, and diphtheria. Endotoxin, it's a cell wall component, LPS. Exotoxin is a secreted weapon. Now we ask ourselves, why can we make vaccines, right? Toxoids against exotoxin, but not endotoxin. Very cool question, because exotoxins are specific protein that can be inactivated into harmless toxoid. We can convert that, but endotoxin are just the structural component, right? So if you mess up the bacteria, you will in fact produce a lot of this wave of LPS, right? So it's much harder to neutralize that way. Let's talk about shape of the bacteria. Super duper important. So we have different type of shapes. Cocci is just round. Bacilli are these rods, right? Spiralas are twisted like a spiral, and most of the bacteria, they fit into this same type. So does the shape is important? Maybe. Actually, I think, yeah, a lot of bacteria, we call them depending upon their shape, how they look like. But combined with arrangement, gram-negative metabolism rapidly narrows the field. Of course, it's like identity card. What about clusters, right? So we have clusters, we have chain, right? Cluster is just groups. Pairs is diplococci, means two bacteria together. Chains is chain of bacteria. For example, this chain, as you can see, the red color. Cluster, as you can see behind the chain, we have this cluster of yellow bacteria going on. This is cluster. And pairs is just cocci, means two, right? Diplococci means two cocci. Let's talk. Why does the grape-like clustering hints at staph? Because staph divides in multiple planes. So daughter cells piles up in irregular clusters, and strap divides in one plane, giving us chain. So strap is a chain, right? And that is streptococci, and staphylococci is a cluster. Let's talk about oxygen classification. Now, where a bug lives in oxygen gradient, this is super duper important, right? So we have three types of bacteria. We have obligate aerobes, right? They need oxygen. Then we have obligate anaerobes. They are killed by oxygen. And then we have facilitative or facultative. They live either way. They can survive in both conditions. And then we have microaerophilic. They want a little bit of oxygen, right? So this is the animation, if you can see, right? We have these O2, aerobes need O2, anaerobes don't need O2. Facultative, they can live either ways. And then we have microphilic, right? And then let's go to the next question. What's the question? Oxygen needs so why it matters? Because of course we are able to predict their habitat and attack them, right? So oxygen need predicts habitat, and the disease microbe cause them. It's like we want to know the location of the culture, right? And we want to treat it. So why do anaerobes thrive in deep absence? Because they love pus and they love dead tissue, right? So pus and dead tissue creates this oxygen-free pocket, right? And this is the exact environment the obligate anaerobes needs to grow. Spores. Now these are spores. These are little children, I should say. So spores are survival pod for hard times. Whenever the environment turns super duper hostile, some bacteria forms this endospores. These are like little spores. And this is a hard shell that resists heat, drying, radiation, chemicals, everything, right? So we have just two big families of spores. We have Bacillus and Clostridium. Spores are why ordinary disinfections is not enough. You need to really autoclave. Now, what is autoclave? Autoclave is just to remove these bacteria using heat. Okay, why does the canned food use such extreme heat? Because of these bacilli and Clostridium, especially Clostridium. They do not die easily without autoclave. So to destroy Clostridium bacteria, we need to boil, right? Boiling is not enough. So that's why low acid canned food are pressure processed, right? So basically the thing is that spores are super duper persistent. They're not gonna die with just simple cleaning, and bacilli and Clostridium are important. Acid fast. Now this is another way of defining a bacteria, right? So there's a waxy coat that breaks the gram stain, right? Now, the bacteria that cannot... Oh, wow, we have an advertisement.
But did you know that there's a YouTube Music app where you...
So, we are back. The acid-fast bacteria. A waxy bacteria that breaks the gram stain, right? So, a lot of bacteria don't sit in both of gram stain, positive and gram negative, right? So, they need a different classification. That different classification is acid-fast bacteria, right? Now, why the gram fails? Why the gram fails? Of course, because their wall is loaded with mycolic acid. So we were talking about peptidoglycan, right? So if a bacteria has mycolic acid inside the cell wall, then this whole gram staining fails, right? So this is a wax, right? That repels ordinary stains. So gram staining is not going to work, of course. So for that, we're gonna use acid-fast classification. So use the acid-fast stain instead, and it holds the red dye, and it is acid-fast positive. For example, Mycobacterium tuberculosis, super duper toxic, right? Why is TB so hard to kill? Because they have these mycolic acid. But the thing is, we have these antibiotics that attack that specific enzyme that is responsible for the synthesis of mycolic acid itself, right? And that's why TB bacteria takes so many months to die, of course. So growth media is just when we take some bacteria out, we put it on a petri dish, we put it on a dish, and we grow it. And to understand what kind of bacteria it is, right? We are trying to guess what kind of bacteria it is. To study the microbe, we grow it on the culture, artificial food like plant soil bacteria grow on agar. Agar is just a food for bacteria. It's a sugar basically, and it forms these colonies. Colonies just their cities, right? So why grow colonies at all if we have microscopes? Because a single cell is invisible to the naked eye, of course. It's hard to test a colony with 1 million identical cells enough to run identification and sensitivity tests on it. Of course, we will understand if the person is sensitive to some sort of antibiotic. If we do the gram culture and we understand, we do the growth media and we test okay, this guy is super duper resistant to that kind. So that's why we have to do it. Now that was agar. Now we have blood agar, right? Now, blood agar, reading how the bacteria attacks the RBCs. We now know, is it alpha partial, is it beta complete, or is it gamma? We have 3 types of attacks on the blood by the bacteria. So blood agar contains RBCs, of course. So we watch the hemolysis, how the red blood cells are getting destroyed by the bacteria, right? So we have 3 patterns. First is alpha. Alpha partial is just partial. Beta complete is 100% RBCs are dead, and gamma is nothing, right? No death. So why is hemolysis useful in first loop? Because the zone type instantly narrows the list. For example, beta hemolysis around gram positive coccus points toward group A strep or strep aureus. So basically, we are trying to identify the type of bacteria depending upon the pattern it creates on the blood, right? So alpha hemolysis, partial destruction, a green halo, right? RBCs injured, not destroyed, leaves a green hue around the colony. Pneumococcus and viridans are classic alpha hemolytic organisms. What about beta hemolysis? So beta hemolysis, RBCs are fully messed up, fully lysed, leaves a clear zone, right? So group A strep, group B, and Staphylococcus aureus are the key beta hemolytic organisms. Gamma, no lysis, nothing, everything is okay. So is no hemolysis still useful information? Of course, because it rules out beta and alpha, yeah, and it points towards Enterococcus. Enterococcus is just stomach and coccus, just telling you. So Meconium agar, very famous. Now this is sorting gut bacteria by using lactose, right? So can a bacteria ferment lactose? If yes, then the color will turn to pink. If no, then no color, it will stay pale, okay? So this is called Meconium agar, and it's useful for understanding your gut bacteria. So pink is E. coli, Klebsiella, pale is Salmonella and Shigella. Why Meconium agar is also selective? Because bile salts and crystal violets in the medium suppress the gram positive, right? And only gram negative gut bacteria appear. Then lactose sorts them by color, right? So we have these multiple ways of sorting these bacteria depending upon the type of agar, right? So catalase test this is also very nice. Does the bubble form or not, right? So catalase test, does it bubble in peroxide? Now peroxide, very important. What's really happening here is that we add hydrogen peroxide and then the bubbles are formed. If the bubbles are formed, it's a catalase positive. If no bubbles are formed, then it's a catalase negative. Now what's the physical relevance? What's the physical thing we are talking about? Hydrogen peroxide is H2O2. Now a catalase separates the gram positive right? A catalase separates the gram-positive cocci into staph is positive, staph is negative. So catalase positive, catalase negative. It's basically catalase test is a test where the bacteria is able to deal with hydrogen peroxide, right? Hydrogen peroxide is basically how your macrophages attack bacteria. Okay, so that's the catalase test. So what are bubbles made up of? Bubbles are made up of oxygen gas. Catalase breaks H2O2 into water and oxygen, the rising bubble that releases oxygen. Coagulase test. Now, does it clot the plasma? Now we add the plasma and clot is formed. That is coagulase positive. So that is coagulase positive. But not a lot of bacteria can do that. So that is Staphylococcus aureus, that it can do it. It can actually create this coagulation of this plasma, but staph doesn't. One test, it's a huge payoff. Yeah, it pulls the dangerous Staph aureus out of the harmless skin staph, of course. Why would a bacteria want to clot plasma? It's an easy question. Because the fibrin clot cloaks the Staph aureus from immune cells, a virulent streak, right? And it's not an accident. It's very super duper easy to identify this using this coagulase positive, coagulase negative. Now the oxidase test. Now we are trying to ask ourselves that is the cytochrome oxidase present in the bacteria, right? So if in this test if the result is purple, then the oxidase is positive. If there is no change, then it's a negative. It helps to separate the bacteria from gram-negative, right? And name a famous oxidase positive troublemaker. Example: Pseudomonas aeruginosa, ear infection. Oxidase positive and notorious case for hospital and burned wood infections, right? We want to do the oxidase test that turns purple. Positive, right? Fungi. We are finally in the fungi region. Now, the fungi have a nucleus, a mitochondria, ER, endoplasmic reticulum, and Golgi, just like humans. They are just like humans. The key difference is that they're cell wall. Humans have none. Humans do not have cell wall. That's why penicillin is okay with us, right? Now, that single difference is where the antifungal drugs mostly target, of course. Because antifungal infection is much harder to treat bacteria. Because fungi are super duper eukaryotic, like us, they are super duper complex, right? So that's why it's super tough to treat them. They always are gonna stay with us. So cell wall, their cell wall contains something called as chitin and beta glucan, and the membrane uses ergosterol. Now this, wow, my ergosterol. So we call it ergosterol or ergosterol instead of cholesterol. Now we can actually give inhibitors of ergosterol, and we can inhibit the formation of this chitin beta glucan cell wall of fungi, and we can actually break these fungi easily. So ergosterol is why antifungal works. Drugs like azoles and amphotericin attack it and spares the human cell cholesterol, of course. Now, of course, we can also give cholesterol inhibitors. Sometimes they do. Why do azoles exploit ergosterol? Because they stop the inhibitor, they stop the formation. They block the enzyme that makes ergosterol. The fungal cell wall membrane fails, and our cholesterol-based membrane are left untouched. Well, what about yeast versus mold? Well, it's just the difference of temperature. Yeast is a single round cell. There's the yeast, and mold is multicellular like this. It has filaments, hyphae we call them. So mold is usually present in cold. Yeast is usually present in heat. So warm is 37 degrees Celsius, tends to create yeast. Cold tends to create a mold. Usually you can see that in fridges, right? In fridges you have moldy bread, but outside the fridge you have yeasty, what should I say? A lot of things are yeasty in hot environment. Okay, why does the body temperature favors the yeast? Well, that's a cool question. So at least at 37 degrees Celsius inside the host, many fungi cells switch to yeast, a form better to of course it's for surviving. I hope you know that. Dimorphic fungi, a fungi that can have it can look in two ways. It can look like two ways, okay? Basically, dimorphic. Morph means how it looks, okay? So dimorphic fungi, one organism, two shapes, set by temperature. So dimorphic fungus changes form. 37 is yeast, 29 is mold. We call it dimorphic simply. Right. Let's go next. Why does this matter? Of course, because we want to keep the temperature low, right? We can deal with the mold, right? So in a cool lab, fungi grow as infectious mold spores. Inhaling them is super duper dangerous. Inside the warm body, they can convert into yeast, of course. What about parasites? Parasites are dangerous. Parasites are big in size. So parasites live inside the host, two families, protozoa and multicellular is helminths. So helminths are difficult to treat. Protozoa are easily treatable. So what makes parasite different from bacteria? They are large, they are eukaryotes, means they are complex, and they have life cycles. And they have life cycle spanning host and stages of their life. So we want to study a journey, right? So the thing is that every parasite works differently. Most parasites are infectious in their spore stage, and then they hijack your RBCs. That's how your malaria works, you know. So that's why parasites are dangerous. Protozoa, these are single cells that move in a different way. Now they have flagella, cilia, and pseudopods. Flagella is a tail, cilia is a hair, as you can see in the animations, and pseudopods is the fake feet. That's the pseudopod. It's my favorite word in biology. So why classify protozoa by the movement? Because it's easier to classify in this way. Their motility organelle is stable, visible feature, right? So we can do it easily. What about helminths? Helminths are the worms, right? And we have three types of helminths: nematodes, cystodes, and trematodes. Now nematodes are roundworms. These are the nematodes. What about cystodes? These are cystodes. What about trematodes? These are trematode flukes. Let's talk. How do tapeworm differ from? Which is tapeworm, by the way? Tapeworm is cystode, as you can see in the middle. So tapeworm is flat, segmented head, segmented, and with the head that anchor to the gut. So this part actually anchors to the gut. And roundworm are super duper smooth, and they are cylindrical in shape, and they are unsegmented. That's a round cylindrical body, nematode. Sterilization, very cool concept. Destroy absolutes everything, right? Sterilization destroys everything, including the spores. Disinfection kills many, but antisepsis is gentle enough for living tissue. So if you want to clean your hands, antisepsis is enough. If you want to go and work in a lab, you want to sterilize everything, including yourself. This is an autoclave machine. An autoclave machine actually increases the temperature at a particular temperature, and at that particular temperature, we will see a lot of sterilization happening, right? So autoclave, ethyl oxide radiation. They are heavy artillery needed to kill resistant spores, right? Spores are super duper resistant. I already told you earlier in the video. Why isn't boiling considered sterilization? Boiling kills most of the microbes, but not the spores, right? That's why. Okay. This had one fact for you. Autoclaving is done at 1 to 1 Celsius under pressure, right? That's important. Normal flora of the body. This is a person, and this person is upside down. This is the head. These are two legs, and cool. Body is an ecosystem, of course. So on the skin, we're gonna have some bacteria. These bacteria are gonna be gram positive. On the colons, we're gonna usually have anaerobes. Mouth, we're gonna have streptococci. On vagina, we will see lactobacilli. That's why it has this smell, right? The microbe protects us, of course, because they are our own ecosystem. They are responsible. They are actually part of our normal immunity, right? So the microbe protects us. Crowding to invaders and shaping immunity, of course, that's very important. So how can wiping all normal flora can cause disease? Of course, broad spectrum antibiotics can clear out protective gut flora, letting C. difficile overgrow, right? And what happens is that a lot of times when you start on broad spectrum antibiotics, you're gonna have diarrhea, because your digestion also depends upon these bacteria itself, right? Your digestion is basically fermentation. Not fermentation, this is a very broad term that I just used very loosely, but your bacteria play a huge role in your digestion, basically. Pathogenesis. Word is that can I enter, can I attack, can I invade, can I evade? So invade and evade are two different things. And then can I spread? Now, why is this framework so powerful? Because we can actually treat the bacteria on different stages. That's why it's so important, right? So immune evasion. Now these bacteria can actually evade your immune attacks, right? Capsule antigen variation, hiding inside the cells, right? IgA protease. Now we have IgA that is a mucosal immunoglobulin, right? and protease means it just cuts these protein, right? So each is a way to survive immune system. Yeah, of course. So why does antigenic variation defeats the immunity? Because it changes how it looks, right? So by constantly changing its surface protein, the pathogen can stay one step ahead. Of course, yesterday's antibody is no longer fit today's code, of course. So that's how it is. So antibiotics works by cell wall, either attacking the cell wall, okay, or they can attack the ribosome, 50S subunit or 70S subunit depending on what kind. Then we can have DNA inhibitors, right? Then we have folate inhibitors, right? Then we have membrane attackers, right? So depending upon cell wall, we have penicillin, ribosome, we have macrolides, DNA, fluoroquinolones, folate, we have sulfonamides, and we have membrane, that is polymyxins, right? So depending upon the antibiotic, depending on the type of thing we want to attack, we can prescribe the antibiotic on that. So this is how the classification of antibiotics is done. And we want to just master these 5 targets, and long drug list are organized. Yeah, of course. So why do wall targeting drugs spares human? We know that answer already. So that's the grand classification actually. So microbiology, is it alive or not? Not alive. Prions, viruses, yes. Bacteria, fungi, parasites. That's it. And that is it. For any bacteria, for any organism, when I ask, is it live dead? Then we can ask, is it prokaryote or eukaryote? Then we can ask DNA, RNA. Then we can ask envelope or naked. And then we can ask gram positive, gram negative, cocci, bacilli, spiral, aerobe, anaerobe, capsule, not capsule, spores, or toxins, right? Endotoxin, exotoxin, which type of toxin? So we don't want to memorize microbiology, we just want to understand it. And then when we understand it, we can start memorizing, because I think this whole subject is just memorization. All right, our course is completed. Have a nice day. I hope you enjoyed this course.